In the previous segment A of Module 2, we talked about the origin of biopotentials. What we want to talk about now is sort of the next step in that. So, biopotentials, as we discussed, originate within the body, usually by some linear superposition of a number of action potentials by neurons or by cells within the heart. Now we have to figure out how to measure that. And so the next step in that is how do you actually obtain the signal? So this particular lecture is taken from chapter 5 of the book edited by Webster. This chapter is written by Michael Newman. All of the slides in here originate from some version of either the textbook or lecture notes that were done using the textbook. So the origin of most of the notes lies within the textbook. So in order to discuss how biopotential electrodes work, we need to go back to a little bit of chemistry and talk about the electrode-electrolyte interface. So the electrode-electrolyte interface, basically you're providing the interface necessary between electric changes in the body and the recording or the measuring device. And this is not a mere metal that conducts electric charges in the body, and so it's not as trivial as it may seem. And we'll look at a number of reasons why during the course of this lecture. So one of the reasons is that the current in the body is carried by ions, whereas the current in the wires are carried by electrons. Another difficulty is that biopotential electrodes contain within themselves some internal capacitance and resistance, as well as the capacitance and resistance that are formed in the electrode-electrolyte interface. And so there must be a function, a transducer function, that describes these biopotential electrodes or an equivalent circuit of some sort. The interface that's required for the charge transfer is the electrode-electrolyte interface. So the electrode is typically some sort of metal with desired properties, and the electrolyte is a conducting media that's specifically formulated for the electrode metal being used. So here's a simple example of an electrode-electrolyte interface, illustrating that within the electrolyte, it is cations and anions that are carrying the current, and within the electrode itself leading to the instrumentation, it is electrons that are carrying the current. And so the cations undergo a reaction where they give up some number of electrons depending on their valence, and the anions end up accepting these electrons also depending on their valence. The electrolyte is an aqueous or liquid solution, and it contains cations of the electrode metal. So you would have electrode metal basically joining the electrolyte solution, becoming ionized as electrons flow away from the interface. So there are a couple of equations shown on the previous figure, that general ionic equations. Now, if the electrode has the same material as the cation, then this material gets oxidized and it enters the electrolyte as a cation, and electrons remain at the electrode and flow in the external circuit. If an anion can be oxidized at the electrode to form a neutral atom, then 1 or 2 electrons are given to the electrode. So this is either oxidation, which is the loss of an electron, or reduction, which is the gain of an electron. So because of this, you end up getting a characteristic potential difference that's established by the electrode and the surrounding electrolyte. And this is going to depend on the metal, the concentration of the ions that are in the solution, and the temperature. It's also going to be dependent on some second order factors that we won't mention here. The half cell potential can't be measured without a second electrode. So if you actually wanted to experimentally measure the potential at the cell, which in order to get a good equivalent circuit representation of the cell, you would actually need to do, then you would need a second electrode. And the second electrode ideally should be a hydrogen electrode because the half cell potential of the hydrogen electrode has been arbitrarily set to 0. So all of the other half-cell potentials are given as a potential difference with the hydrogen electrode. The reason that you have a half-cell potential is that you have charge separation at the interface. So these oxidation or reduction reactions that occur at the electrode-electrolyte interface give you a double charge layer, and this is actually quite similar to what happens in a semi-permeable membrane in a cell that passes anions and cations back and forth. The one difference is that you don't have the movement of ions in the electrode, you have the movement of electrons. Nevertheless, you still end up with this double charge layer, positive on one side, negative on the other, and so this gives rise to a potential. So here are some examples of just different half-cell potentials. As you can see, this was published in 1975. Very little has changed since then. This was the work of early electrochemists. Um, you can see that different metals undergo different kinds of reactions. Here is silver, which is a common one. Silver chloride is an extremely common electrode material. And you can see what the reaction is, and then you can also see what is the half-cell potential. So this half-cell potential appears like a DC source in a circuit model that would be done of the electrode-electrolyte interface. And so now we need to talk a little bit about polarization. If there's a current between the electrode and the electrolyte, which you would expect there would be if there are potentials that you're trying to measure, then the half-cell potential is often altered due to polarization. So an overpotential is a difference between the observed and the zero current half-cell potentials. And there are some things that affect this. First of all, there is the potential difference due to resistance. When you have current flow, that changes the resistance of an electrolyte, and therefore a voltage drop results. There are concentration effects, so changes in the distribution of the ions at the electrode-electrolyte interface contribute to this overpotential. And then finally, activation. The activation energy barrier depends on the direction of the current, and this is going to determine your reaction kinetics. So the net over potential is the sum of these three effects. Of these effects, the polarization and the electrode impedance are the two most important electrode properties to consider. So like the situation where we had um, a permeable membrane or a semi-permeable membrane, we have an electric potential that exists. And that was defined, as you recall, by the Nernst equation. So in this particular case, where you have ionic activity at the electrode, you have this standard half-cell potential, and then you have this term that's a function of the concentrations of the different constituents in the oxidation reduction reaction. Now, in this class, we're not going to get deeply into the chemistry of half-cell potentials and electrochemistry. So my goal in showing you this slide is not that you would memorize the Nernst equation, but that you would get an idea that basically what's going on here is that what happens at the electrode is a function of the electrode material, which is where you get your half-cell potential. It's also a function of temperature. It's a function of the materials that are involved and their relative ionic concentrations, as well as the number of valence electrodes, which is also a function of concentration. So the little a's are basically ionic activity, which is the same as concentration. So this gives rise to the fact that there are two different kinds of electrodes. There are polarizable and non-polarizable electrodes. So perfectly polarizable electrodes. These are electrodes where you don't actually have charge crossing the electrode-electrolyte interface when you apply a current. And so basically the current across the interface is the displacement current. In other words, we know when we have a capacitor that if you put this capacitor in a circuit, you can calculate a current through the capacitor. However, what the current's actually a result of is it's actually a result of the time-varying charge distributions that occur along the capacitor plates. So in the case of a polarizable electrode, the situation is similar. you have a charge distribution, and so the current that flows is not current that's actually crossing this gap. It's current because there is a time varying charge distribution at the plate, and so electrons and or ions must flow in order to facilitate that charge distribution. An example of a perfectly polarizable electrode is a silver-silver chloride electrode. And typically these electrodes tend to be used for recording. So in my lab we have a number of silver chloride electrodes that are used with my EEG system. We also have other newer types of electrodes. But in the conventional, especially clinical situation, silver chloride is one of the dominant electrodes used in making measurements. The other kind of electrode is the perfectly non-polarized electrode. Now these are electrodes where current passes freely across the electrode-electrolyte interface. So you don't have this sort of energy threshold that has to happen in order to make the transition. These electrodes don't see any overpotentials, and so an example of that would be a platinum electrode. Typically, these electrodes are not used for acquiring data. They tend to be used for stimulation. So if you've ever had an injury and you've had to have E-stim done, then probably you had a perfectly non-polarizable electrode applied in order to deliver that E-stim. Platinum is a common source for this electrode and or a common material used in stimulation electrodes, non-polarizable electrodes. So here's a illustration of a silver chloride electrode and just an illustration of what happens when that's placed in an electrolyte containing chlorine atoms. So what happens is you have this layer here at the surface that forms. So you have a silver metal, you put that in a solution containing chlorine atoms. The positively charged silver ions and the negatively charged chlorine atoms form a surface layer of a salt. The human body contains, as you know, a lot of sodium chloride, a lot of potassium chloride, and so there's a lot of chlorine present in anything that you would use to model um, the human body in human cells. Here are some other examples. This is a silver chloride, sintered silver chloride. So this one is formed by a mechanical process as opposed to a chemical process. There's a lead wire that's attached to the electrode that is basically a silver wire that minimizes any interface issues between the wire and the electrode itself. So here's the equivalent circuit for the biopotential electrode that's in contact with an electrolyte, where EHC, this voltage source here, represents the half cell potential. R subscript D represents the impedance that's associated with the interface and any polarization effects, as does C subscript D. And then R subscript S is the series resistance associated with interface effects and due to resistance in the electrolyte. So this would be your circuit model for the part of the probe, not the transducer itself, but from the actual transducer over here on the left would be sort of the electrolyte surface, the electrode surface. This would represent all of the interface effects associated with the contact. So we're going toward the body as we move right, and we're going toward the instrumentation as we move left. So we find that impedance of the electrode is a function of frequency. So this is an electrolytically deposited silver chloride layer on silver electrodes with an area of 1/4 square centimeter, which is not an unreasonable um, set of numbers. And then you can see impedance is measured as a frequency of hertz, and you can see that for various different manufacturers, we definitely see a decrease in impedance as we have an increase in hertz. Now, many of the body signature waveforms lie between 0 and 100. Another sort of subcategory would be between 0 and 5K. You can see up here at the high frequencies, this is sort of leveling out. This is a log plot. So if you're measuring an EEG, for example, and your frequencies of interest are, from 1 to 50 hertz, then if you were to draw this on a linear plot, you would see that they're relatively, it looks pretty flat. Here is the magnitude of the impedance as a function of frequency for electrodes in general. This is experimentally determined, so you can see that you're relatively, once again, magnitude of the impedance relatively flat until you get to about 100 hertz, and then you can see you have something that looks like a low pass filtering effect. However, that does tend to level out here at the higher frequencies. So it's not a true low pass filter. If it were a true low pass filter, you would continue to have this cutoff in the transition region. But we do drop by 2 orders of magnitude over 2 orders of magnitude of frequencies. So this is representing sort of a first order filter, which we would expect because it can be modeled as an RC circuit. So the next thing to think about is that to the right of our little equivalent circuit exists the human body. And so now we have to figure out how we make contact to the human body and what the human body's sort of equivalent circuit looks like. So you have your electrode electrolyte interface, and then you have the skin interface. Now I want to pause here for just a moment and explain that typically when electrodes are placed on the skin, there is some sort of solution, either a gel or a saline solution, that forms the connection between the electrode and the skin. So when we're talking about an electrode electrolyte interface, that interface is frequently the conducting gel, not the skin surface itself. So you need to be sure that you're aware of that. All right, so the skin consists of 3 layers, the epidermis, the dermis, and the subcutaneous layer, and we are most interested in the epidermis as that's the main place that contacts the electrode. This is referred to as the stratium corneum. This is where skin cells go to die. And then the layer just below that is the stratum granulosum, and then just below that is the stratum germinativum. And this is where new skin cells are formed. There are also, within deep layers of skin, some vascular components and some nerves, as well as sweat glands and sweat ducts and hair follicles. So with the exception of sweat glands, we don't really have any particular characteristics that affect electrode performance. Hair can be an issue. So a lot of times for really good measurements, the region where you're placing the electrode might be shaved. People have gotten aware they're pretty good at doing high impedance amplifiers. So in other words, an amplifier that will handle a high impedance electrode. So that is not the problem that it used to be. But it's still, if you're having trouble acquiring good data, there's always the possibility of removing the hair as well as removing the outer layer of dead skin and skin oils with just a very mild, like abrasive treatment, almost like you do if you've got a facial. So that's the skin interface. Here is a figure of what we just explained. The electrode would sit here. You can see there's lots of stuff going on. You've got some nerves in here. Here is the hair follicle cell and the hair root. Here is a sweat gland and a sweat pore. And then as you go on down, you can see there's some blood here. As you know, if you prick your finger, it's going to bleed a little bit. So you've got some capillaries and some little capillaries here, and some small blood veins that exist in the skin layer. Got veins and even arteries that can exist in that area just below the skin. This is just another view. Here are the 4 principal cell types in the epidermis. What you typically try to do when you apply an electrode, or what you used to try to do when you applied an electrode, was just to wipe off this outer surface of skin Even a rough Kleenex is more than adequate to, so just a little tissue usually gets quite a bit of these outer. And what you're concerned about is how flaky this is. So you either have to have a good kind of gel that will penetrate down into these flakes and fill these pores, or else if you think about it, your electrode that you sit on top of here is going to only make contact at certain spots. And also the dead cells, they don't have the electrolytic um, properties that a living skin cell would have. This is just an actual photomicrograph of a portion of skin representing the actual um, presence of these layers. So the model of the electrode skin interface, now we want to include the skin. So here is your half cell potential. Here is your capacitive and resistive polarization effects. Here is the series resistance, and notice that the series resistance sort of incorporates the gel that is between the electrode and the outer skin layer. Now here's your outer skin layer. So there will also be a half cell potential between the outer skin layer and the gel. So you actually have two half cell potentials, one between the electrode and the gel and one between the gel and the skin. That's represented here. Um, these are equivalent capacitances and resistances that are actually due to the epidermis. And then in parallel with that, because they have very, very different properties, is the capacitance and the resistance and the half cell potential due to sweat glands and sweat ducts. So these can be kind of an issue because depending on how many sweat glands and sweat ducts you have and how much you're sweating, the signal can be very different. In fact, one biological measure that's often done is to measure what's called skin galvanic um, properties. So you're actually measuring how much people are sweating. At one time, that was used in lie detection. It's been proven to be not incredibly accurate, but people do still monitor that, not on its own, but as sort of an array of biopotentials and biometrics, biomarkers. And then finally, you have your dermis and subcutaneous layer, which sort of plays the same role as the gel. It forms just a resistive path through which current can flow. Here's an example of the skin impedance for a 1 centimeter square patch at 200 kiloohms, or 200 kiloohms at a hertz, 200 ohms at a megahertz. So the skin impedance is much lower at higher frequencies. So motion artifacts occur because electrodes move, and the charge distribution when electrodes move gets screwed up. And so when the charge distribution gets screwed up, the signal goes wild. When the signal goes wild, the measurement goes off the window, and of course, the most extreme case is if the measurement goes off the window, the patient dies. These are from, this little slide is from a guy named Ravi Polakar, who was at Roland University. You can find it on his website. You can find it on Roland's website, and I like his sense of humor, so I included his slide on motion artifacts. Motion artifacts indeed are a problem because as your electrode moves, you do indeed mess up your charge distribution. And so that causes then signals to appear that aren't representative of biopotentials, they're representative of motion. When you use non-polarizable electrodes, this reduces the low frequency noise that's created by motion artifacts. And so that's why it's very important to use ECG and EEG to use such electrodes. Now, when you're measuring an EMG, it's not as important because typically EMG signatures are higher frequency signatures. And so you don't have, and they're almost all differential. And the differential elements are placed close together. So often, if one sensor has a motion artifact, both sensors have a motion artifact. However, it's also possible the gel skin contact can create motion artifacts. So just using silver and silver chloride doesn't always eliminate all the problem. And the issue is that motion artifacts tend to be low frequency, and in the ECG and the EEG, the actual signal that we're trying to measure is in the same frequency band as the motion. So here are some examples of biopotential electrodes. This is one that if you've ever gone into the clinic and had someone do an ECG, that's what these typically do. There's a kind of a foamy surface. It's got some adhesive on it. There's then a metal disc. Um, it's kind of like um, I don't know. It's kind of like a snap. If you've ever had like a snap on your clothing, the top of the electrode has a snap that connects to the conducting wires. Um, Usually, the top of the electrode is reused. The bottom is disposable. B here is just a metal disc electrode that's applied with surgical tape. Um, and A is a metal plate electrode that can be used to apply to your arms and legs. I've never seen this used. This figure shows up everywhere. This electrode shows up everywhere. I've never actually seen this used. I've never even seen one or seen one for sale. Same here, suction electrode, sometimes used in clinical ECGs. Never seen this used either. The contact surface is here. Lead wire terminal is here, and so the suction is what's going to hold it in place and apply some pressure to hopefully reduce movement artifacts. This is a common one, the recessed electrode with the top hat structure. This is fairly common also in EEG. You have a little insulating package. Um, this area in here gets filled with gel. The actual electrode contact is at the top, and these are reusable. This is what we actually, if you go and a person does not use a cap to do an EEG, they will use these little electrodes. And then number C is a disposable recessed electrode. It's kind of the same general thing as B, but often made with a less expensive material. And you can see there's a gel-coated sponge. Snap is coated with silver and silver chloride. So once again, all of these are gel type materials. They're all silver, silver chloride because of the superiority of that in a recording electrode. These are some flexible electrodes made to be worn while you're moving, or if something is moving. They tend to be, if you have to wear something for 24 hours, these tend to be a little more comfortable. And then sometimes, in order to avoid the whole skin problem, you actually use percutaneous measurements, meaning inside the skin. So these are little tiny needle electrodes where the needle actually penetrates the surface of the skin, goes into the muscle. And then you can do, the advantage of this is you can do a highly localized recording right in a particular part of the muscle. The disadvantage is obviously it's invasive, and so that can be painful, and it can also hold with it the possibility of introducing an infection. You can actually make internal measurements in the brain as well. There are implanted electrodes that go within the brain and make measurements. Typically, these are used in very extreme cases, and often they're used if you're trying to ascertain either a very specific focal point of where a seizure is occurring so that you can kill that part of the brain to eliminate the seizure disorder, or you're using the electrode for stimulation in the case of someone, for example, who maybe has some kind of brain damage and you're trying to do stimulation. But even that stimulation has now become more of an external process. Here are some examples of microfabricated electrode arrays. So these are silver, silver chloride. It's on a chip. It comes out to a series of contacts. Here's another geometry. So all of these would be where you're basically making multiple measurements. So the electrode itself is going to have its own equivalent circuit. Here's an electrode that's being tested in a physiological saline solution. So what they do is they design their electrode and then they use a saline solution because that's probably the closest thing in percentages, that's the closest thing to represent body fluids. Um, then you can sweep this as a function of frequency and actually get what these numbers look like, or what the output versus input looks like, or the log of the impedance magnitude versus the log of frequency. You can get that as a function of frequency experimentally. So this would be what an equivalent circuit looks like. If you now start to change that as a function of frequency experimentally, you can derive. And so you can see this is similar to the figure that we saw, that as frequency increases, the magnitude of the impedance decreases until it reaches a relative constant. So that's the pretty much summary of biopotential electrodes. Um, Not all measurements use biopotential electrodes. There are some that monitor things um, in other ways. Um, there's a move to non-contact monitoring of things like respiration and heart rate to look and see if a person can use different kinds of electromagnetic radiation to probe the body and actually use things like Doppler shift and other uh, techniques to determine what the heart rate is. That's currently an area of research. The thought being that in situations like a very crowded ER, to be able to monitor vital signs without having to actually go and apply an EKG might be really useful. So the biopotential electrodes are generally used in a research or a clinical environment where the patient is stable or relatively stable and the instrumentation is available, and tends to be pretty standard what's used clinically. What's used in a research lab um, has advanced considerably, but FDA approval always lags research development. So that's the state of biopotential electrodes.